The previous introductory video on proteins explained the different levels of their structure and the different roles they play in metabolism of living organisms. This video will take a more in-depth look at protein structure, namely protein motifs. This is important because the diversity of protein function in facilitating metabolism arises from a surprisingly simple principle: their structure. The way proteins are built determines their role in the cell. As mentioned in the previous video, these biological polymers are made up of long chains of amino acids coded for by DNA nucleotides in a gene. Each amino acid sequence is unique, leading to a vast array of protein structures. The two key events in understanding protein structure, its linkage to protein function and metabolic role, were first, the pioneering work of Frederick Sanger in sequencing the amino acid structure of bovine insulin in 1953. The next breakthrough occurred in 1960 when X-ray crystallography enabled the first determination of the 3D structure of a protein. As more proteins were sequenced and scientists delved deeper into the intricate world of proteins, they began to notice recurring patterns in their structures, which sparked a new wave of curiosity and research. Certain combinations of amino acids, arranged in specific three-dimensional shapes, appeared again and again in different proteins, often performing similar functions. These patterns were not random, but highly specific. These recurring structural units were termed as protein motifs. The first, helix-turn-helix, was discovered in 1982. Researchers highlighted their fundamental importance to understanding protein function. Think of protein motifs as modular building blocks, like Lego bricks that can be assembled in different ways to create a variety of structures. Each motif is like a unique Lego piece with its own shape and purpose. They can be categorized as lying between the secondary and tertiary structures of proteins. Combinations of secondary structures, such as alpha helices and beta sheets. Each motif has a characteristic shape and function, and by combining these motifs in different ways, nature creates a vast repertoire of proteins with diverse functions. This modularity allows for incredible versatility in protein design. These motifs are not merely decorative elements, they are essential for the protein's function, playing critical roles in the protein's activity. They can act as binding sites for other molecules, provide structural support, or facilitate chemical reactions, making them indispensable to the protein's role in the cell. The world of protein motifs is incredibly diverse, with hundreds of different types identified so far. These motifs are fundamental to the structure and function of proteins, playing crucial roles in various biological processes. Some of the most common motifs include alpha helices, beta sheets and beta barrels. Each of these motifs has distinct structural characteristics and functional roles within the protein. Alpha helices, as their name suggests, are spiral-shaped structures that resemble a coiled spring. These helices are stabilized by hydrogen bonds which help maintain their shape. They are often found in proteins that span cell membranes, acting as channels or transporters. These channels facilitate the movement of ions and molecules across the membrane, essential for cellular function. Beta sheets, on the other hand, are flat sheet-like structures that resemble a pleated sheet. These sheets are formed by hydrogen bonds between strands of the protein. They are often found in proteins that provide structural support, such as silk and keratin. These structural proteins give strength and flexibility to various biological materials. Beta barrels are formed when several beta sheets curl up to form a barrel-like structure. This unique formation creates a hollow center, which can be used to transport substances. These motifs are often found in proteins that transport molecules across cell membranes. They play a critical role in maintaining the cell's internal environment by regulating the passage of substances. These are just a few examples of the many different types of protein motifs that exist. The variety of motifs allows proteins to perform a wide range of functions in the body. Each motif has a unique shape and function, contributing to the incredible diversity of the protein world. This diversity is what enables proteins to be so versatile and essential to life. There will be a future video on protein domains. If you liked this video, please press the like button and subscribe to this channel. Also press the notification bell to be alerted to future videos published on this channel. Thank you for watching and stay curious.